Hello and welcome to the January edition of the LSCAT Speaker Series. I'm very excited, we're all very excited to have David Morrissey here with us. He is a PhD at Pfizer and has spent decades in the fields of molecular and cell biology, studying nucleic acid and gene editing based therapeutic approaches. And so most exciting for us today is he is going to talk about what is new, the latest and greatest all about CRISPR. And so this isn't just CRISPR as some theoretical off in the future thing. This is what's going on with it right now and how it's going to impact the next generation of drugs and life-saving medicines. So David, thank you so much for being here. And would you like to get started and tell us a little bit about your work?
Sure. Let me share my slides. Okay. So what I plan to do is really focus on therapeutic applications of CRISPR today and start off with some basic background, history of the technology, how it's evolving, and then highlight several clinical applications, programs that are in the clinic, and showing some positive data. So it's an exciting time in the field. And as you'll, I think, note as I go through the presentation, there is certainly some overlap in technology with the mRNA vaccines. mRNA can be used to express Cas9, and LNPs can be used to deliver the CRISPR machinery in vivo, and that's actually being done in the clinic with successful early phase readouts. So it's an exciting time, and one can see now sort of the generalities between the various RNA technologies and how development of some of those early modalities, such as siRNAs, has really impacted the speed at which the CRISPR technology has moved forward. All right. So just, if anyone's curious where the name comes from, it's not about making CRISPR lettuce, but I'm sure that is an application that's in the works. But CRISPR stands for Clustered Regularly Interspersed Short Palindromic Repeats. So say that 10 times fast. So this really dates back to the first sort of notice of CRISPR. And that was back in the 1980s when scientists were starting to sequence bacterial genomes, and a series of repeats were noted in the E. coli genome. And they were curious, but there was no idea at that point what they were there for. More and more sequencing in various bacterial types really led to further identification of these repeat regions. And finally, in the mid-2000s, around 2005, a group of food scientists working in industry, led by a scientist by the name of Rodolphe Brangu, who worked on yogurt cultures and developing the bacterial strains for yogurt cultures, really figured out that CRISPR was an innate immune system that protected bacteria from viral infection, bacteriophages being the viruses that infect bacteria. So that was really one of the major first steps in understanding that. Led to a paper in Science that got some attention. And then a couple of years ago, two scientists who were at Northwestern University at the time, Eric Sontheimer and his postdoc, Luciano Marfini, determined that the target of the CRISPR system was not RNA. In fact, people thought, well, this must be like RNA interference in bacteria, with the RNA being the target. They identified DNA as the target. And I should note that those three scientists were all turned out and became founders of Intellia Therapeutics, one of the first generation CRISPR companies that I was honored to be one of the first employees of. And then it was really in the 2010 to 12 timeframe that Jennifer Down at University of California, Berkeley, and her collaborator, Emmanuel Carpentier, really start to understand mechanistically how the CRISPR system worked, and then start to apply its use to eukaryotic cells. So with some relatively minor modification of the system, we're able then to apply it in targeting eukaryotic DNA in a target specific manner. And that, I think those three groups of scientists and those three events really led to, the CRISPR revolution now, and all that's happened really in just the last 10 years. It's really astounding. We've gone from understanding how the mechanism works to developing as therapeutics and seeing positive clinical readouts in about 10 years. And I think the reason this has moved so fast is all the prior work that was done on RNA-based therapeutics and delivery. So there is a huge diversity of CRISPR systems in bacteria, and I won't even begin to go through this, but what we know is CRISPR is largely centered around Cas9, which is a type 2 system, but there are multiple systems. And some bacterial strains have, each cell contain multiple systems. The biology around the innate use of CRISPR is fascinating in bacteria, and how the bacteria acquire sequences from the bacteriophage, integrate those into their DNA, and then use those to surveillance system against infection. Fascinating biology. We won't be covering this today, but there's a huge effort to continue to mine bacterial strains for new CRISPR systems that could be then developed for applied across a variety of fields, including therapeutics. And behind Cas9, there are other Cas systems like Cas12 and Cas13 that are currently being applied to therapeutic use. And this is an analogy I really love, and that is the Swiss Army knife with CRISPR. When you think about it, at the heart of this, and what's really being used is a system that recognizes DNA in a sequence-specific manner, and then can be modified to impart that different functions can be imparted on that basic system to modify DNA in various ways. So, the current generation that's in the clinic basically cuts double-stranded DNA, and that causes indel formation, insertions or deletions, or one could use that to insert a new gene. But on top of that now, fusion proteins can be made with Cas9 and a variety of DNA modifying enzymes that can impart epigenetic modification, DNA methylation or demethylation. Instead of cutting DNA-based modification can be done with deaminases. So these are all now being developed behind the basic CRISPR system, and I think we'll see these going into the clinic in short order. So as I said, I'm going to focus on therapeutic applications, but to begin with, CRISPR has really changed how one does biology, how one does research. And in the course of my career, the last 30 years or so, there have been several really fundamental discoveries that have had the same type of impact. PCR polymerase chain reaction to be able to amplify DNA was one of those that have had broad applications in research and diagnostics. And of course, I think everyone, if you weren't familiar with PCR-based tests, you are now with COVID. And then the second one was RNAi, RNA interference, the ability to silence gene expression with short RNAs. And that in itself has led to a whole new class of drugs over the last few years that are on the market in treating patients with rare disease. And CRISPR is being applied to various forms of bioprocess, biotechnology, the food industry, agriculture, livestock. It's and this is now. I mean, there are. I don't think people recognize, but there have actually been CRISPR modified yogurt cultures and yogurt on the market for a number of years now. And then the therapeutic applications, which, I think we're just starting to see the tip of the iceberg, and I'll talk about some of those today. So the CRISPR 1.0, really, there are, the idea CRISPR was going to solve everything. Ultimately, it may, but I think it's also important to understand what it can do and it cannot do. So in its original form, Cas9 cleaves double-stranded DNA. And what happens next really is dependent on the cell and the state of the cell and how the DNA repair machinery comes in to repair that double-stranded break. So in the simplest iteration, and this is what we're seeing move into the clinic first, is just disrupting gene expression through indel formation. So CRISPR-Cas9 cuts double-stranded DNA in a site-specific manner, and then that's repaired, and typically that's repaired with a small insertion or deletion that then disrupts the expression of that gene. And we'll talk more about that. One can then use pairs of guide RNAs to excise a region of the DNA, so deletion. And I'll show you an example of that. And then one can insert a new copy of gene. One can make a double-stranded break with Cas9 and then insert a new copy of the gene. And we'll see an example of that as well. So just a few terms up front that we'll be covering, and science, like a lot of fields, there are a lot of abbreviations, a lot of acronyms. I tend to use those, so I want to make sure I cover a few of these up front. So as I already said, CRISPR clustered regular interspersed palindromic repeats. That's a great piece of trivia that you can pull out with your friends. So the Cas proteins, these are CRISPR-associated genes or proteins such as Cas9, Cas13, Cas12, etc. The single guide RNA, or sgRNA, this is what really imparts a large part of the sequence specificity of CRISPR. The PAM, or the protospacer adjacent motif, I'll talk more about, but that is the sort of the sequence element, recognition element within the protein itself, in addition to the sequence recognition element that the single guide RNA imparts. And then thinking about what happens once Cas9 cleaves the DNA, double-stranded break, or DSBs. And we'll talk a little bit more about homology-directed repair versus non-homologous end joining. And then, as I said, indels, which are known as insertion deletions. And then in terms of delivering the CRISPR system, there are two main ways: lipid nanoparticle, which most people are now quite familiar with and have actually been dosed with the COVID mRNA vaccines. And then AAV, which is adeno-associated virus, which has been used extensively in the gene therapy field, and in some cases has been used to deliver the CRISPR system clinically. So this is the basic schematic. So showing the Cas9 protein in blue, the target RNA, and then the single guide RNA. So, thinking about the sort of the invention of CRISPR and Cas9, I think one of the major inventive steps has really been the single guide RNA. In the inherent bacterial CRISPR system, there's two parts of the guide RNA. There's the CRISPR RNA, which contains the target sequence. In the bacterial system, that's known as the spacer. And then a tracer RNA, which is sort of the universal RNA, and that these two interact through base pairing. And Jennifer Doudna at University of Berkeley, who won the Nobel Prize last year for the CRISPR system, along with Emmanuel Carpentier, she put these two RNAs together with a stem loop, and which really, helped facilitate this in terms of convenience. At first, there was a challenge to synthetically make a guide RNA of about 100 base pairs, but when there's a commercial driver in science, that often gets things done. And now making a 100 base pair guide RNA by solid phase synthesis, RNA synthesis, is routinely done. And that guide, in some settings, is chemically modified for stability and abrogation of the innate immune system with modified bases that have been used in other similar modalities such as siRNAs. And I think it's important to note that this is much like the mRNA vaccines. They're also chemically modified for the same reason. So it's stable and limiting the innate immune response by cellular sensors such as toll-like receptors and RIG-I. Cas9 is been engineered in a variety of ways. The PAM, this is the recognition system that each Cas9 has its own PAM sequence. So for it's a standard Cas9 that everyone is using really in the field comes from Streptococcus pyogenes, also known as SpyCas9, and its PAM sequence is NGG. But through standard protein engineering approaches, the PAM has been altered, so almost any PAM can be recognized by an engineered Cas9. There's also been versions of Cas9 engineered to have higher fidelity, so reduction of off-target cleavage has been achieved this way. Cas9 nickase, so the cleavage which really has just cleavage of one strand versus two strands, that allows other approaches such as base editing, and then orthologs of Cas9 from other bacterial species such as Streptococcus aureus have also been used. Some of these Cas9 orthologs are smaller in size, which has enabled delivery by AAV viral vectors, which have size limitations. And we'll talk about one such case going forward here. So this is it's a modular system. It's also highly engineerable, and people have tapped into other Cas9 orthologs from other species. Some of them are smaller, but don't have the same level of efficiency of cutting, but may have other uses going forward. So as I said, there's a huge effort underway in the field to mine bacterial genomes for new CRISPR systems. So as I said, the Cas9 will cut double-stranded DNA in a sequence-specific manner based on recognition of the PAM motif and the target sequence from the single guide RNA. And what happens next is really up to the cell. And so the most likely outcome is that the non-homologous end joining system causes the formation of an indel. And based on the state of the cell, whether it's cycling or post-mitotic, that indel can be quite small in post-mitotic cells, such as hepatocytes in the liver, or it can be broader, and that can be more difficult and challenging to control in terms of off-targets and larger chromosomal rearrangements that can be deleterious. The goal here has been really site-specific repair, and that's been more challenging. And this uses the homology-directed repair system. This requires a repair template. The repair template can be a wild-type allele if it's a heterologous mutation. The wild-type allele can be used as a repair template, but the frequency of this is really quite low, and it's been challenging to improve the frequency to the point where it's going to be clinically, therapeutically viable. What appears to be really more viable is sort of the insertion, and I'll talk about that, where one can supply an insertion template to insert a new copy of the gene. And I'll show you an example where AAV, the viral vector, is being used to deliver an insertion template, which is inserted once Cas9 cuts the target site. So in terms of therapeutic application, there have been two general approaches. The first is ex vivo, in which cells are removed from the patient. They're engineered using CRISPR system and then returned to the patient. And in this case, really the product, the drug product is really the engineered cells. And I'll talk about two general applications. One is in the immuno-oncology field, engineering T cells to attack tumors in a target specific manner. And the second is engineering hematopoietic stem cells to correct diseases such as sickle cell anemia. And this is actually in the clinic and is really showing positive results. And then the second approach, which has been a little bit slower to get to because the technical challenge, is delivering the CRISPR system through systemic administration. And the examples are in the clinic. This is intravenous administration. And in this case, which has moved in the clinic first, is using lipid particles to deliver, and Cas9 being expressed from the mRNA. So much like the COVID vaccine technology that many of us have been dosed with. And I have to say, now that the ex vivo approaches are starting to work, the next generation, I think, of those will be to convert those to in vivo delivery, being able to use lipid nanoparticles to deliver to stem cells or immune cells directly through systemic administration. Because as we'll talk about briefly, the ex vivo, the regimens required to harvest cells from patients are really grueling. And if one can avoid those and target cells in vivo with CRISPR, it will be much easier on the patients. So I'm going to talk just quickly about in vivo immunotherapy for cancer with CAR T cells. So these are T cells that have been engineered with a chimeric antigen receptor. And this work was really pioneered initially by Carl June at the University of Pennsylvania, really pre-CRISPR. So CRISPR is not necessary for doing this, but it's really facilitated the sort of next generation approaches in a way. So in this case, T cells are removed from the patient. They're engineered to express a specific receptors that recognize a tumor, and then they are put back into the patient. And for some patients, this has been completely game-changing, and it's really sparked the whole revolution in immunology-based approaches to cancer. And now, what the next generation here, those are called autologous T cell therapies, where the patient's own T cells are harvested, engineered, and put back into the patient. The next generation is called allogeneic, so off-the-shelf CAR Ts, where T cells are engineered to be given to any patient. And so this is where CRISPR really comes in. So using CRISPR to knock out genes that are involved in recognition of self, non-self of the T cells. And then it's much easier to insert a CAR construct into the genome of T cells using CRISPR. So this is really the next generation that is still very challenging, but there's much effort being put into this field. So, but I want to spend a little bit more time now on ex vivo CRISPR therapy for both sickle cell disease and beta thalassemia, which are related diseases. And this is caused by mutations in the beta globin gene. And so this is in the beta subunit of hemoglobin. And if a patient has are homozygous for mutations in the beta subunit, that leads to sickle cell disease. And if they are heterozygous, that mutation, that's beta thalassemia, which is a form of anemia. So, I mean, this is a large number of patients worldwide with these diseases. And, this has been, I think, game-changing. I mean, certainly bone marrow transplants were being used in the past, but now CRISPR-based therapeutics of correction are really starting to show utility in the clinic. So the basic scheme here is that there infants, there's a separate form of hemoglobin. So in the adult, there are two types of subunits, the alpha and beta subunits. In each complex, there's two subunits of each type. But in newborns, there is the beta subunit hasn't been turned on yet, and a gamma subunit is used, which is ultimately silenced by a repressor element in a gene called BCL11A. And what the idea with CRISPR is to target the BCL11A, the repressor within BCL11A, to edit it out and then upregulate fetal hemoglobin, which then replaces the mutated beta hemoglobin subunit. And as I mentioned, hematopoietic stem cells are collected from patients. This is really a grueling regimen. It's basically bone marrow ablation, and then those are engineered. The BCL11A locus is edited with CRISPR, and then the cells are put back in the patients. Now, there's several different companies have clinical trials running right now. I'm going to show you some data that comes from CRISPR Therapeutics. Novartis also has a program in the clinic right now. I don't think they've reported any results, but the interesting aspect about the Novartis program is they had a compound already in clinical trials that expands hematopoietic stem cells in culture. So what they were able to do is to isolate cells, edit with CRISPR, and then expand those cells, which means that they was more likely that the graftment will take just based on the cell number. But so as of last summer, CRISPR Therapeutics had treated more than 45 patients in this effort. And I'm just going to show you one piece of data. And what to mention on the left-hand side shows baseline data from a patient population, where at that point they were seeing very low levels of fetal hemoglobin, HbF, about 4%. And what you see is that post-implantment of these engineered hematopoietic stem cells over time, the amount of the fetal hemoglobin increases up to about 45%, and stays stable. In this case, at least for, this is data, I think it's probably almost a year out. More than a year. This is months after. So 21 months out, these patients have stable levels of fetal hemoglobin at levels of about 40%. So this is amazing result. This is really going to be curative to these patients. And this is still an early clinical trial, but it's looking very promising. So here's, I think, really the first example of a CRISPR-based therapeutic going in the clinic and showing positive results for patients. And again, putting this in perspective, this is really just about 10 years after, when there was enough of mechanistic understanding of CRISPR to think about applying it to engineering eukaryotic cells. So I think an astounding rate of progress in the field. Okay, now I'm going to shift gears and talk about in vivo approaches to deliver CRISPR systems in patients by systemic administration. So there's been two basic approaches used. One, lipid nanoparticles, again, much like the COVID mRNA vaccine, and adeno-associated virus, AAV, which has been used extensively in patients for gene therapy, for delivering a gene that is missing or mutated in a patient. So there's some pluses and minuses to each approach. I will be quite clear that I'm a proponent of the LNP approach, and was fortunate to be involved in what's really become the first clinical trial of LNP administration, in vivo administration of the CRISPR therapeutic. So some of the advantages are transient expression of the CRISPR components, Thinking about this, we're delivering a bacterial-derived protein into humans. Bacterial-derived proteins are typically immunogenetic in humans, and Cas9 is no exception. So one doesn't want the protein expressed for very long. Also, in terms of the potential for off-target effects, modification of other sequence besides the target sequence, one wants to minimize by time of exposure with the CRISPR system. It's also then redosable if one needs to dose up over time to a certain level of editing in patients, or do maintenance doses if the effect starts to wane based on cell growth or organ growth over time. One can maintenance doses. And there's low immunogenicity to LNPs. There's not much in terms of immune recognition. The downside right now is there's limited tissue tropism of LNPs. So LNPs really have been applied in two ways clinically previously to this. One is in the vaccine space by intramuscular injection, and the other is delivery of siRNAs to the liver. And so the first therapeutic siRNA that is now on the market, clinically approved by Alnylam Therapeutics in Cambridge, which targets transthyretin gene, which we'll talk about in a minute. That's delivered by lipid nanoparticles. That's very similar to what's being used for CRISPR. But beyond that, LNPs go to the liver for two reasons. One, obviously the liver is a filtration organ, but secondly, they really select it for interaction with circulating lipoprotein complexes that contain the ApoE protein. So LNPs interact with the ApoE protein containing complexes, and then are taken up into hepatocytes to interact with the LDL receptor. And other cell types actually express the LDL receptor, so one can see lower levels of uptake in those types of cells. But largely LNPs have been liver-centric. But over the last few years, there's a lot of effort to expand the tropism of LNPs to other tissues by new components, new lipids, and new compositions that are, there's some compelling data with LNP delivery to spleen, lung, and to some extent muscle cells now. So I think we'll see the space opening up greater in terms of what tissues can be delivered to and then treated with CRISPR-based systems. AAV has the advantage of expanded tropism. There are different AAV serotypes that have different tropisms in the body, and those have been utilized to a great extent. The real issue with AAV is that there's immune response to an antibody-based response. And so patients can only be dosed once, because if they're dosed again, there'll be antibody response that will neutralize the effect of the viral vector. And in fact, many people have pre-existing immunity to AAV. It's a pretty promiscuous virus. So not every patient can be dosed with AAV effectively. So that's been a downside. And the field is working on ways to be able to redose AAV, but a solution has not been really found yet. The other downside for AAV for use of CRISPR is its persistent expression. So that means Cas9 is expressed continuously over time, probably for many years. That ups the chances of immunological response to it and increases the chances of off targets. So the one case I'll talk about with AAV use in patients for delivery of CRISPR is direct injection into the eye. So local delivery into the eye. And the eye is more immune privileged. So far, this has not seemed to be an issue. Patients have not been redosed, but there hasn't been any kind of dose-limiting toxicity based on immunological response to the viral vector. And then a number of payloads can be delivered, especially the LNP is really versatile in terms of what the cargo can be. It can be plasmid DNA, and that's been largely used more in a research setting. And what's really being used mostly is Cas9 mRNA and a single guide RNA together. So this just shows really sort of the timeline again, and really driving home that how quickly this has happened. So the first reports of LNP delivery of CRISPR with successful editing in mice was reported in 2017. There were a couple of papers out that year, one of which I was a senior author of, showing that we could knock out expression of the transthyretin gene in the livers of mice, and that reduction in TTR expression was stable for at least a year in mice, which was a result that surprised us, actually, that we thought with, hepatocytes do turn over, liver regenerates, hepatocytes do turn over to some extent, but that really meant that we must be hitting a stem cell population that as new hepatocytes were being produced, they had the knockout of the TTR gene. And then this quickly moved to into editing in non-human primates. And while that data from Intellia, I don't think was ever published, but, by 2018, there was proof of concept that this could be done in non-human primates. And now in 2021, the first report of successful systemic delivery and benefit to patients. So I won't spend a lot of time on this, but delivery is challenging. Delivery for every RNA or nucleic acid modality used therapeutically is very challenging, and one can start to think about the number of steps in terms of targeting of tissue, uptake into cells, endosomal release, trafficking to the nucleus. There are a lot of ways where this can go wrong or not work efficiently. And in reality, the most inefficient aspect of delivery is really in the cell. And I think there's still much room to improve these systems, and there's much work underway. Safety, of course, is an issue in terms of potential dose toxicity and immune responses, both antibody responses and innate immune responses, cytokine storms generated by that innate immune response. As in LNP and RNA, it looks a lot like a virus, and the cell can respond with innate immune response, but through chemical modification of the RNA and selection of components of the LNP, this has been really mitigated. And as I said, tissue and cell type tropism specificity is still a work in progress, but we're seeing continuous progress in the field in this regard that will open up editing to additional cell types and tissue types over time. So we're running behind time here. So I'm just going to quickly, this is Editas Medicine, a local company in Cambridge, that they have, this was the first clinical trial where patients were dosed directly with CRISPR systems, and this was for eye disease. This is, they're targeting a gene called STEP290. This is a mutation in this gene causes a form of retinal degeneration, and they're editing out the mutation with CRISPR, and they're using AAV5 to deliver this by direct injection into the eye. And one thing to note is that they use Staph aureus Cas9, so an ortholog that is smaller than spy Cas9, and they're able to fit this into an AAV vector that has limited cargo capacity of about 4.5 kb. And in this case, what happens in STEP290, there's a mutation that causes a cryptic exon that causes premature truncation of the protein, and using Cas9 and 2 guide RNAs, they're editing out that cryptic exon to express a protein that's then functional. And they've reported phase 1 results where they show this is safe in patients. They haven't reported efficacy yet, but Typically, phase 1 trials are really focused on efficacy with the potential on safety with the potential for efficacious readout. What I really want to talk about is then this in vivo editing program from Intellia. This is to edit, again, this is again LNP delivering Cas9 expresses an mRNA and a synthetically modified guide RNA. And this is treating a disease called transthyretin amyloidosis. Transthyretin is a protein that's expressed in the liver. It carries thyroid hormone and vitamin A, the protein in some people becomes misfolded, and that causes a form of amyloidosis that affects nerves, heart, kidneys, and eyes. Up until recently, there really wasn't a treatment. As I said, actually, the first siRNA drug on the market is to treat TTR amyloidosis, and that's showing great utility. The idea behind the Intellia program was that with a single administration, patients could be essentially cured of the disease. So this is clinical results that they reported out last summer. So patients have been treated, and there are 2 dose levels here, but this is an astounding result. This is looking at levels of the TTR protein in circulation in these patients. And it's essentially knocked down to about 95% of wild type levels. And that's sustained out for a year with a single administration of the CRISPR therapy. So this has been game changing for the whole field. And this really opens up now in vivo delivery of CRISPR systems to treat patients directly. So again, this is astounding how quickly this has moved. For example, Intellia Therapeutics was founded in 2014, and here, 7 years later, is an astounding positive clinical result. So the field is moving at an amazing pace, and patients are already being positively impacted by CRISPR. I want to show just one other piece of data approach that I think this is cool. This again is from Intellia, and this combines both the LNP system to deliver the CRISPR components, but then using AAV to deliver an insertion plate to insert a new copy of a gene. And so this has been done in non-human primates at this point. It hasn't gone into patients yet. And this is to treat a disease called alpha-1 antitrypsin deficiency, and it's to correct a mutation in a gene called SERPINA1. I won't go into details of this, but what I do want to show you is, again, this is in non-human primates where using the system of LNPs to deliver the CRISPR system, AAV to deliver a new copy of the gene. It's been inserted in the albumin locus in hepatocytes, and this shows clinically relevant levels of the alpha-1 antitrypsin gene in non-human primates with a single administration of this therapy. The duration is at least a year that this new copy of the gene is being expressed. So again, I think this is an astounding result. This is likely to go into the clinic shortly to treat patients. And then, since we're really running short on time, I'm going to just quickly say a word about what's coming next in terms of genome editing 2.0. And as I mentioned up front, CRISPR system Cas9 and the other Cas proteins that are being utilized can be really seen as a platform to add additional functionality. One can make a mutant form of Cas9 that doesn't have cleavage activity and use that as sort of the sequence-specific recognition element, and then add other protein functionality. And this can be base editing to change the identity of a base through deamination. And there's a company in Cambridge called Beam Therapeutics. They're working on this, and their first program was also in sickle cell disease. I think that's getting ready to go into the clinic. Some feel that this approach may be safer because there's not double-stranded breaks that could lead to larger genomic chromosomal rearrangements. There's also epigenetic editing coming, where one could use methylase or demethylation of epigenetic marks on chromosomes to impart turn on or turn off a gene permanently. So, this is just, I think, the tip of the iceberg, and we'll see more approaches as one can take those sort of what now are old school approaches for DNA modification in a test tube, the old days of cloning, and apply those to the CRISPR system and do this in a cell and do this in an organism and do it in a patient. So I will leave it at that and happy to take any questions.
Thank you so much, David, for a glimpse of what's happening right now and what we have to look forward to. I definitely have a few questions. I know that we're bumping up against 45 minutes, but I think that these are some interesting points, and there's a lot to unpack here. And so you reviewed a lot of current therapies that are currently in the clinic, and big if here, but if progress continues and if things keep trending in this direction, how many therapeutics might we have by the end of the year?
Well, behind those that have gone to clinic, each of those companies I mentioned have a pipeline of other projects. So, for example, Intellia, based on success, early success with the TTR program, have started another clinical trial for another rare liver disease. Because the system's modular, you just swap out the guide RNA to target another gene, you use the same Cas9 mRNA, the same LNP, and that really speeds drug discovery. So, Intellia has a pipeline of other programs behind that, which can move really quickly by just, identifying new guide sequences for a particular target gene.
And when the technology is scaled up sufficiently, could this potentially provide cost-saving measures, since it seems relatively easy to just swap out versus formulate,
Yeah, and I think what we've seen with the COVID mRNA vaccines, which one of the questions has been, the ability to scale the technology, manufacture it at a large scale. That problem has been cracked, and I think that's one of the real amazing feats of the vaccine effort for COVID is the scale at which these vaccines are being produced. Pfizer, is producing billions of doses per year now, and no one ever envisioned mRNA and LNPs to be dosed at that level, manufactured at that level. And of course, once you get to that scale, the cost comes down. So absolutely, the modularity, and now that manufacturing at scale has really been cracked, I think the field will move even faster.
And by 2050, what % of all hospitalizable diseases could potentially be treated by CRISPR?
Well, there's a huge number of genetic diseases, and a lot of them are rare diseases where, the patient population is so small that there's not much of a commercial driver for drug discovery around those indications. But, the modularity of the system will allow, I think, really potentiate development of therapies against those rare diseases. And that's where really seeing CRISPR applied first is these are all indications with relatively small patient populations. And, once the technology is proven in those patient populations, we'll see it applied to larger diseases as well. There's literally hundreds of rare diseases that can be treated.
Two more questions. One is, what is the moonshot that all the companies want to get to?
We just saw the moonshot. This was the moonshot 7 years ago. So I guess the question is going to be, what's the next moonshot?
What's your personal?
It's come to fruition. It's working.
Yeah. What's your personal moonshot?
Well, focusing on delivery, expanding tropism, increasing specificity. The sort of the holy grail would be cell type specific delivery. And so, yeah, there's a huge amount of work going in the field, and that's one of my real personal interests.
You actually answered my last question regarding what are you most excited about with CRISPR? So yeah, thank you for sharing that. Faith, is there anything you have?
Yeah, I've just got one question, kind of stepping back a little bit and thinking about our audience for this talk being a population of science teachers who are educating the next generation of scientists who will be taking up the mantle of this work that you're doing and this, the next generation of science discoveries. So I'm wondering, what sort of attitudes and skills and ways of thinking would you encourage our teachers to be nurturing in their students?
I think a lot of people, at least in the general population, think of science as sort of a static body of knowledge. But science is a process, and the process of doing science, and it's ever changing, ever moving forward. And I think the general public has gotten a bit of a taste of that with COVID. Seeing the sausage making, that it's open source. Some results stand the test of time, others don't. They fade away in terms of reproducible science. So I think there's a lot of creativity involved in science that sort of the general population doesn't see. And work like this, molecular biology, cell-based biology, and engineering of technology is really a creative process. So I feel fortunate in my career I've had this really interesting mix of some basic science. Think about the basic science of CRISPR or basic science of RNA interference, understanding those mechanisms, being able, based on that mechanistic understanding, apply technology development to develop those as therapeutics, and then see it go into the clinic to help patients. So it's creative and it's rewarding. So fostering creativity and being open-minded, and science is fun. Okay, this is a blast. Being able to do this is just so much fun. And then there's a reward, seeing patients benefiting from it.
Thank you,
Awesome.
David.
And it also sounded like, from a lot of your stories of different therapeutics and different timelines of these discoveries over the last several decades, that there's an immense amount of collaboration both within teams and among and between teams. And I think that that's something that I do see a lot of our science teachers bringing into their classrooms is teamwork, but I think there's always room to get better at really teaching those skills.
Exactly. Science is a team sport, and that's how it's approached in industry. Teams within a company or teams with collaborations. Part of my role at Pfizer is external collaborations with both biotechs and academic labs. And exactly, I think being able to work in a team setting and bringing people together with different disciplines really moves the science much faster and makes it more fun.
Totally. Thank you so much, David. I think your talk has provided so much richness and nuance that can help our teachers who are implementing CRISPR labs in their classes to be able to really enliven that for their students and bring it to the sort of real world. So thank you so much for taking the time to talk with us today.
My pleasure. It's been fun.
And high school students love hearing about how the technologies are actually being used right now. And the fact that it's all happening for folks watching in the Boston area, this is happening miles away from us. So it's really cool to think that the next generation of scientists could be anyone doing this lab right now.
Absolutely. Yeah, I mean, the biotech here in Boston, pharmaceutical industry, it's become a big driver of the economy. And there's no place on the planet like Cambridge. It's with such close proximity, and it's a really close-knit community because of that. I have to say, working from home, I haven't missed being in the office so much, but I miss being in Cambridge and being part of that community.
Totally.
Well,
awesome. Well, thank you so much. Michael, do you have any closing words?
Nope. Thank you so much for watching, and yeah, have a great night.
Thank you. Thanks, everyone.
Take care.